We will continue with this, and I will. I like to show the final some of the final results, so I might go a little bit fast and explain the conceptual part rather than going into details. But as you can imagine, there is a lot of complexities and difficulties when feeding the data for predicting complex traits. Ordinary least square, usually they don't work. There is this we call the course of dimensionality. So we use this bias regression. We use a penalized linear regression. We ended up with the rich and the G-BLUP. We know in genomics, many important traits and diseases are highly, well, they have intermediate to high heritability, many low heritability. So maybe we don't need so many individuals and so many markers for the traits which are highly heritable. Yeah, we may or may not. It depends, because I talked to the pathologists in CIMI. They say, well, no, but this is horizontal resistance, this is vertical resistance. So we don't know sometimes the number of markers, the number of genes. And we have, on the other hand, this modern genotyping sequencing method that can describe the genome very precise. So we have the genotype, the phenotypes, we make the prediction models, we predict and we make decisions. These are the phenotypes, these are the genetic values. The genetic values are related to the markers. The X are always the markers. So we usually what we do is just to use something, a function of the markers. And the way we put the markers into this G sub X, Gx, is the model. And then we have a residual. The complexity comes because there are so many markers. We don't know which markers, what type of interaction, dominant, epistasis. We have here environment, so we have markers, environment. So there is a lot of complexity, big data sets, a lot of information, and pedigree and markers, phenotypes, and that is the complexity. So I will talk a little bit of this, and then I will jump to the other presentation and show something on G/E, and then show some results. On the regression, we'll consider the number of parameters exceeding by large the number of data points, at least in genomics. In this particular context, ordinary least square, maximum likelihood, sometimes cannot be used because they don't offer a unique solution. So we usually use a type of a penalized regression, which involves either we shrinkage the estimates and we say, I will take only small piece of pizza, very small. Or I start deleting markers, right? So I delete you, and you give me all the pizza, and I eat all the pizza. That's it. It's something, well, not myself, maybe 2 or 3. This is a variable selection. You select the markers. The thing is that when we have, so either this shrinkage estimation are not very precise for estimating the marker effect, that's for sure. But they are very precise when you add their effect, when you add them up for prediction. They are very precise, but not for estimating the marker effect. So all these regression, shrinkage, they are not for estimating the marker effect. They are not for that. That's the reason why doing a GWAS with a rich regression is a really bad idea. But you can do variable selection. You can use a statistical model that will say, I'm going to take only this marker, that, and that. That is all right. But then you need to see which one of those, and then you need to see if those are related to real QTLs or no. So it's always problematic. We prefer not to do anything, just to use the penalized regression, and that's it. The two most important challenges are the complexity we have to incorporate in our models, the complexity of the genetic mechanism that might be involved, and solving this course of dimensionality. Because also in ordinary least square, we assume that the predictors are independent, which is not the case for markers, because the markers are linked. So they are not independent. They are not, they are related. Therefore, when you try to find the inverse of the matrix, you don't find it. You cannot. The metric is singular because the markers are related. And that's another problem. So that's how do we confront this? Well, this is the standard regression model. I will skip all this, and I will show you
so the slides will be available.
Yes. Yes, you have it there. I like to show you these equations because those are the rich regression equations. If you look at this equation here, x prime x, if you ignore this lambda d, if you ignore this and put this is equal to 0, this is the solution, or these are the ordinary least square solution. On the rich regression, we add a quantity here. Why we add a quantity? Because by doing this, the thing is that this matrix x prime x is singular. It's singular because there are the predictors are not independent, they are related. So this x prime x, this matrix of sum of squares and relationship, this is a sort of covariance matrix. By adding this in the diagonal, we try to diminish the importance of the off diagonal that gives you the relationship and gives this x prime x matrix singular. We sort of decrease that importance. We add a small quantity to the diagonal, and then we find, and we can find the inverse of that by doing this sort of trick. But this is formally, statistically, this is very formal, because the problem is an optimization problem, and this we minimize. This is the sum. This is the residual sum of squares that we minimize in the ordinary least square, but we add a quantity here. And so this is what the rich regression does. And it works very well. And but the estimates may not be unbiased, they might be biased. But they are very precise when you add them up. So this is the beta rich regression. I will go fast on this because you don't need to. This is the rich regression BLUP is the estimation, the prediction of the BLUP, of the main effect, using the rich regression, using this equation that will put on the diagonal something there, this lambda, which is a ratio of variance component, and will make this x prime x non-singular. Now, how do you go from the R-BLUP to the G-BLUP? Well, this is the equation. This is a standard Meg-Wiesen equation with the linear model. These are the marker. These are the marker effects. These are the marker values. And you can put this into this equation in metric notation, right? And this X is n by p. P is the number of markers, which is usually standardized for the ridge regression. Beta is a vector of unknown marker effects. And we assume that the residual has a normal distribution. Yes?
How do you standardize that when you say standardize?
Oh, the marker effect. Just standardized by the center and standardize.
Okay.
Center and standardize. Assuming you have filtered the markers for the non-monomorphic markers. Non-monomorphic means that you markers which have the same value, they don't discriminate anything. You usually delete them because they make the model more parsimonious, because you don't need to estimate any effect for markers and they are all 0, 0, 0, 0 set for 1 or 2 individuals. Or those which are 1, 1, 1, 1, there is no need for doing that. So we center and standardize the markers. Now, so if this happens, if beta is assumed normal with 0 sigma square b, this is the R-BLUP. If we make this transformation and we call u is equal to xb beta, then equation 1 is represented by this other equation, which you have u here. And u is the vector of random genetic effects and independent from the residual. And u has this normal distribution with the G matrix sigma square u, and the G matrix is x prime. Then you say, what's the difference between x prime x and x prime? X prime is n times n. X prime x is p times p. So if you have 1 million by 1 million, I wish you luck if you want to invert that metric. And by n is okay. Still is a huge one. And this has been proved by Van Raden. So the solution to this equation is the G-BLUP. The other one is the R-BLUP. And this is what we usually do most of the time, not every time, in genomics, because this is very simple and everyone understands this and use it. So equation 1 and 2 are equivalent. We can, and also in the previous equation, we can have a fixed effect here. You know, if we have sites, you can have site as a fixed effect right here. Or you can put any other effect coming from the design, replicates in complete blocks. Okay, so this, I wanted to say this briefly for the R-BLUP and the G-BLUP, because I wanted to show you now what some of the results we have done for G by E, and then go directly to the results of CIMIT for prediction. So, genomic prediction with G by E, we have done a couple of models for incorporating G by E. There are basically some of the models we work. Others are the models that Nicholas Heslott and Mark and Jean-Luc have developed. also using environmental covariables. So those are the two types of G/E models that we've been using. So the genomic and the pedigree-based linear reaction norm model, is a model that has been used in animal science, the reaction norm model. And then these two models, this and the marker by environment, those are very similar to each other. They are not equivalent, there are some differences. But this is a marker by environment interaction. This is a genomic model. Then I put here hybrid prediction, we are not going to see anything like that. The threshold model, we are not going to see anything like that. Usually we have big data. We have genotyping by sequencing technology, highly dimensional data. We might have environmental covariables collected. We have pedigree, we have markers. How do we put this into a model that will account for this random variability? This is an extension of the G-BLUP. And we introduce variance structures that are function of the marker genotype and the environmental covariables. And these are the models. I'll show you in an easy manner. This is a standard model that the breeder has. The effect of the I genotype on the jth environment is equal to the mean, a general mean, the effect of the environment, the effect of the lines, and the interaction line by environment, and a residual. If we have genomic markers, genomics, the G metrics, we can incorporate the G metrics into the model in this manner. So instead of having the L, we might have the G. The G is representing the L. The G will explain part of the L, maybe not all of them, but part of that. The L is just the lines, the entries, one number, 1, 2, 3, 4, 5. The G represents, it qualifies the entries and say, no, this particular entry has this marker structure. This other entry has this marker structure. And this marker structure is represented by the G matrix that has n by n, all the individuals with all the markers. We can do the same with environmental covariables if we have collected environmental covariables. We might have maximum temperature, minimum temperature, the precipitation, and all these, and use it. Instead of saying environment, we characterize the environment, and we say, well, this particular environment is the experimental station in Sydney, this part in this part of the crop, and this is the maximum temperature, this is minimum temperature. We characterize environment. We don't say this is Sydney experimental. This is not Elbatan experiment. No. We say this is an environment, and we characterize the environment like that. So we can do similar to what we have done with the genomic covariables, we can do it with environmental covariables. And then we can also sort of characterize the environment by line, doing the interaction between the genomics and the environmental covariables. It's a little bit complicated, this, but you trust me that this can be done. And
Are you going to talk about how you choose your environmental covariance?
No, but we can talk about this because I think this is important. We usually have maximum, minimum temperature, the precipitation, growing degree days, but I don't know what others can be used. I don't know if you have some other ideas.
Do you synchronize the environmental covariates with the maturity of the different varieties?
No, we say, for example, we say this is a planting day, this is more or less the flowering. But sometimes it's difficult because we don't know, because the trials, Ravi plants the trials at different times. So. Especially when you have a lot of trials. When you have only one trial, you know more or less when, but we struggle with that. And that might make a difference. In CIMID, for some reason, we didn't get much influence on the environmental covariables. In Obregon. I don't know why, because in Obregon is, experimental, the data is there. I mean, they collect it there. It's not data that is collected 10 kilometers away. It's right there. But we haven't been able to explain much. The environmental covariables have not been able to explain much of environment, as we have the genomics or the pedigree with this. Maybe we need to synchronize more, but we don't know. We need to think about it. But this is important. Now, how much of the G/E might be explained by the interaction? Due to imperfect LD between the alleles and the markers, and alleles at the QTLs, marker may not fully account for genetic difference between lines. That's clear. Environmental covariance might not fully account for difference due to environment conditions. Therefore, the proportion of G/E might not be fully captured in the interaction genomics by environmental covariables. That's clear, at least for some of the data that we have. For this experiment that we did with data from Arvalis, because Arvalis was interested in developing these models, we did find some good response. We have in this particular data, we have 340 locations by year combinations. We have a total of 130 environmental covariables describing the environment's conditions. The phenology of the crop was divided into 5 phases. Lines were genotype with a SNP, and the trait analyzed with grain yield. And so these are the models that we fit: environment and line, environment and genomics, environment, line, environmental covariables, and environment, genomics, environmental covariables. These are main effects models, and here we put the G/E. Here we put the genomics by environment, and here we put the environment, genomics, environmental covariables, genomic by environmental covariables, and G/E. And we tried this model with the 2 cross validations. We use CB1 and CB2. CB1, you take certain number of lines, you take them away from all the environments you have. For example, line 3 and 1 and 5, out. We predict them. The CB2 is an easier one. We take some lines from some environments, others from another environment. These are the predictions. These are the models. These are the estimates for CB1. These are 50 random partitions eliminating 20% of the lines across all environments. This is 0. The genomic start predicting something. This is environment line. This should be line. Environment line and environmental covariables. You see, the environmental covariables did not help much, but genomics and environmental covariables did help something, but not much than this. Now, when you put G/E, you start improving something. This is genomic by environment. This is genomic by environmental covariable, and this is the full model with environment, genomics, environmental covariables, G by environmental covariables, G/E. This is the CV2. When we predict some lines in some environments, others in others, the prediction is easier. So I think that with this, I will, for this particular topic, I will stop here. I want to give you this Harvest Plus with data from BLUP and Ravi. They have grain, iron and zinc on the grain. These are the models. These are the predictions in different sites. You see that the predictions are for the genomics are this level for each of the sites. The sites, this is Mexico, Oregon, 2 years. This is India, the WITS Research Directory, and this is Punjab. So you see that the full model increased the prediction for zinc on the grain, for CV1, for CV2 more, and also for iron. So these are very good results that I guess that we are still working with BLUP to show that this can be predicted very well and can be used in genomic selection.
So I have a question. In
Yes.
Genomic selection, do you guys use a set of bytecode populations or just a random germplasm?
Yeah.
And if both, which one is more accurate?
Okay, yeah, that's a good question. No, in wheat we don't use biparental. No. In maize, yes. In wheat, no. In wheat, I guess the idea or the tendency is just to predict advanced lines in different environments. This is important because you can do genomic selection under what is called rapid cycling, which means you are in an F2 population and you predict the progeny with markers, and the markers will do everything for 1 or 2 cycles. In wheat, the wheat breeders are very efficient in making selections at the F2, F3 level, right? So, and economically, this is not possible, just to genotype 1 million plants. So that cannot be done. So in wheat, the tendency, at least in semi, is just to make the predictions of the advanced lines. That is different. It's like a sparse testing. You evaluate something here, and you predict this there. Okay, so I'll finish here this topic. You have the slides here. I like to show you some results, because all this that we have done, it is important, but this is not real genomic selection because we didn't do it. We just take a data set, did the random cross validation, and say, this is good. The prediction is this. For this trait is this. For that trait. But we didn't do any prediction, real prediction. We didn't do any rapid cycling. That will be done by the breeders, not by the...
Is that Arvalis data set publicly available?
I don't think so. I don't think so, no. It might be now. After 2 years, it might be. I need to check with them, because it's a good data set.
Yeah, I'd be interested in finding out who the contact is.
Yeah. It's what's the name? No, now that guy is not there anymore. But I can find it out. Yeah, because that was a constraint we have when we submitted. But that was 2 years ago. So genomic prediction and selection in seed, wheat, and maize. So in wheat, your question is exactly that. we predict advanced lines in the F5, 6, based on G by E. Why not in F2? Because large population size, very effective visual selection, very high cost of markers. I mean, you don't do this. You don't do this in wheat. In maize, because in maize at the F2, they cross the F2 with a tester, then the prediction can be based on G-BLUP doing rapid cycling, and I will show you some results. I know that in CIMMYT, being in a wheat or been sitting with wheat breeders and talk about maize is almost, you are finishing CIMMYT. No, well, I'm just kidding. If you are with wheat, you talk about wheat. If you are with maize, you talk about maize. You never talk maize with wheat, with maize, except we in biometric. No, but I think that is interesting to see the results of maize. Why F2? Because, well, not many markers are needed, and the F2 will have maximum LD between the markers and the QTL, so you have this a very great opportunity to do a good prediction and a rapid cycle. So these are, so the USDA project with Jesse Poland and Jessica and Ravi and Enrique and Julio Huerta and Suji Smita. This is a big data set because Jessica decided to go 5 years back, or you or Ravi or who decided that? But that's a good decision anyway. Go 5 years back and get 50,000 genotypes lines and see and genotype many of them and look at the phenotypic data and try to do some predictions using pedigree and markers. So we have the pedigree, the coefficient of parentage, we have the markers, we have the phenotypic data, we have a total of 49,000 lines with COP and a total of 20,000 lines with markers. The main idea of the project is just to be able to predict lines in India, but also lines that are in the international testing trials in different mega environments, at least 2 optimal mega environments, ME1 and ME4. ME4 is drought? Or no? Yeah, it's drought, right? Yeah. So those are the idea, but basically in optimal environments. And in Louisiana, Jabalpur and Pusa, in 2 years, there were about 500 lines. So this slide from Jessica, incorporating, this is more or less the breeding program in CIMMYT. You have the cross, you have the selected bulk via the shuttle breeding, Obregon and Toluca El Batan. You have advanced breeding lines, you genotype and predict yield under different environments. Here we have about 10,000 lines in optimal environments, and then you select 1,000 and you put it in 5 environments in CIMMYT, which is optimum, heat, drought, severe drought, and sun management, zero tillage or something. So this is the advanced yield trials, and then from here you send it to the 3 sites in India, and some of them go to international nurseries. So the idea will be just to predict these advanced lines within CIMMYT, but more important in India, in these 3 sites in India, and also in the international testing nursery. So we can predict the performance of the lines there. Here we have 50, 60 entries here that we would like to predict. Here on the Indian sites, we have about 400, 500. So, and then we have the yield from South Asia, that is incorporated into the prediction models and back to Mexico for prediction. So making prediction of advanced inbred lines, that was the idea in this project. We have phenotypes for the last 5 years, genotypic data for 40,000, genotypic data for 9,000. Genomic prediction, we have pedigree prediction, so we have the genomic prediction on this side, the pedigree prediction on this side. We predict based on genomics, based on pedigree, and we take the average.
What are you averaging, the GBBs?
Yes. Yeah. These are the estimated realized prediction accuracy for yield in different environments. We have like 0.4 in optimal, 0.15 in drought, late heat, 0.4, severe drought, 0.3. Now, I like to show the predictions in these 3 sites. In Louisiana, 2013, the prediction was 0.46. Did you see that? Yeah. 0.46. In Jabalpur, 0.2. In Pusa, 0.14. Louisiana next year was 0.44, a good prediction for Louisiana. Jabalpur was a little bit higher in 14 than 13. Pusa a little bit higher, 14, 16, over 13. I would say these predictions are not bad.
What's the phenotypic correlation between sites in, say, an Obregon versus
Was about, I don't remember. I don't remember if I calculated or not. But they should be around no less than 0.5. Phenotypic. Genotypic, I don't know. And then also we did the opposite. We take the data from the guys in India and we predict Obregon. And the prediction was 0.37. So the question is, yes?
Do they use the same set of material to predict phenotype or not?
Yes, yeah. Yeah, the same. This 780 something. 300, well, depending on the site, 379, 400, yeah.
Is there any relationship between the training population and the predicted population?
Some relationship? Yeah, yes.
Between the training population.
Yeah. Right, because those are coming exactly from this part. The guys in that the GIDs that were tested in India are coming from the 10,000 yield trials. So there should be a relationship. So the predictions, these predictions, they don't look bad. This is the reverse. This is the prediction of using the training, the guys in India, and predict the performance of the optimal size in Obregon. A little bit lower than the previous one. So the data in Obregon seems to be still I guess Obregon seems to be still a good site for predicting some sites. What I need to do, or what we need to do, is just to make the predictions for the international nurseries, and I have data here. This I need to do. But here there are so many sites, like 10 sites in optimal environment, 10 or 5 sites in drought. I need to do that and see. I give you a brief description on the results on genomic selection in maize in Africa. In Africa, Rex Bernardo Massman and other guys and Rex Bernardo were the first one in 2008 to show that in one biparental population A by B, they did a rapid cycling of selection using markers and show genetic gains. Joseph did this for how many populations? We did this for 8 populations of tropical maize, evaluated in drought and optimal environments. We have this cycle 0 to 100 individuals in drought and optimal. Then we cross cycle 1, then cycle 2, and cycle 3, and we evaluated cycle 0, 1, 2, and 3. They did what a breeder will do. Will take individuals from cycle 1, 2, and 3 and evaluate them. These are the results. This is across 8 populations, grain yield in tons per hectare. This is the cycle 0 result, cycle 1, cycle 2, cycle 3. This is days to mature, days to antithesis. This is plant high. This is for 3 DTMA populations. This is for 5 WEMA populations. Those are different populations that they use. So there are 8 biparental populations. So these are the gains after the third cycle, the second cycle of genomic selection. Actually, from here to here, this is phenotypic selection. From here to here, genomic selection. From here to here, genomic selection. The same for these 3, the same for these 5. They compare this with the pedigree selection, with the conventional selection. And then they have the F1, the cross, and the parents and the checks. But you see that there is, I don't know if this is significant or not. This is the LSD. They might be significant, this one. This, cycle 3 and cycle 3. There is a gain. For example, for this population, across these 3 DTMA populations, this is the gain per cycle after 2 cycles of genomic selection, in average 69, under drought conditions, 69 kilograms per hectare, per cycle under drought.
How long is each cycle?
In a standard one in maize, 4 years. So they were able to use about 2 cycles per year, which is a time. I mean, if you do, phenotypically this should be like this, the line should be steeper, but this is not bad. This is for 5.
Saying the phenotypic selection would have better gains?
Genotypic selection will be better per unit of time. Per unit of time. Per year, not per cycle. Per cycle, no. But per year, per unit of time, yes. Because you divide this by how many years are there. This is for the WEMA population. The WEMA population has a decrease in cycle 2 and increase in cycle 3. Under drought, Under drought. Now, so this is for each of the population. There are 8 populations here. You see, for some populations, cycle 3 did not gain much. For others, actually nothing. For others, increase a lot. For others, also increase. For this, increase. For this, cycle 2 was better. For this, cycle 3, and so forth.
Why are some of the cycle 1 lower than cycle 0?
I don't know. That's a good question. It shouldn't be like that. Maybe they screwed the phenotypic selection.
Is there a way to know which population will be good and which will
No, no. I don't know. We don't know yet. Maybe Rex Bernardo knows. We don't know. Because maybe it's related to the diversity of the parents. I really don't know.
I think at least sometimes it's related to population size and the variation in the population.
Yeah. That might be the case. But they tried to put here, they tried to use 200.
They did.
Yeah. And the same number of markers, like 200 markers, 200. Because I remember I did the prediction using just a standard G-BLUP.
And how many parents were recombined in every cycle? How many
Yeah.
parents were
Right, yeah.
Well, how many were recombined?
Yeah.
I don't remember how many.
You don't know the selection intensity?
No, about 10%, I guess.
So 20.
But I'm not sure. I need to read the details because this has a lot of details on how this was done and all this in order to make it more. Actually, Joseph tried to compare it with pedigree, with the conventional, and it's not a fair comparison because for doing that you need to have the same number of lines and all this. Now, I'd like to show you something very, very interesting because I think that this is something that goes a little bit beyond what we are talking. So these are another set of biparental populations in optimal environments. You see that cycle 3 is ahead of cycle 2, a little bit, not much. Cycle 1, cycle 0. Well, the big jump is cycle 0, 1 because this is phenotypic selection. Then here, not much increase in cycle 2, cycle 3, yes. These are the F1s, these are the parents, this is the pedigree conventional, and these are the checks. So this is the for the optimal environments. Cycle 0, cycle 1, cycle 2 the same, cycle 3 increase. This is in drought environments. The line is not that steep than for optimal environments. But this is very interesting. What they did was this. They developed lines from the marker selection. They developed lines for the standard conventional selection. And they tested these lines. They test the lines. The hybrid developed from the lines from the marker selection, then from the conventional selection, and they put it in yield tries in different environments to see if the hybrid from the marker selection or the genomic selection, they were superior than the hybrid from the conventional breeding. These are the results. These are the means of hybrid from C3, from the parents conventional cross checks in 2 biparental populations evaluated in 3-4 drought, 3-4 optimal. This is standard multi-environment trial analysis. These are the top 10 lines, I guess. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The top 10 lines were always for marker-assisted selection or for or for genomic selection. These are the grain yield. This is optimal for optimal environments. Again, all the top 10 lines were from genomic selection. These are the parents, the crosses, the conventional, and the checks. For this population and for this population, the top hybrids were coming from the C3 genomic selection. Very interesting. Very interesting to see the stability and then the high yielding of these lines here for this particular population. And these are 2 or 3 sites under drought, 2 sites under drought, or 3 sites under drought. You see, the black ones are the lines, the hybrid developed from the lines from marker selection. Always the best hybrids were coming from the lines of hybrid selection, on marker selection. For drought, always these black entries are coming from the hybrid from marker selection. Then the F6 are from pedigree, the white one are from the checks, and you see this consistently. The same in optimal environments, consistently in every population, the best lines, the best top lines across all these optimal environments, they are coming from the marker selection. This is remarkable because this is like this is the end of the story. They say, well, you know, we did the rapid cycling, we developed the lines, we crossed the lines with the tester, we did all this in a multi-location testing trial across environments in drought and optimal, and these are the results. This is exactly what the breeders do under conventional selection. But they show that this, so they can say, well, now we can say that the hybrid developed from the lines on marker-assisted selection, they work all right. So the conclusion, at least for maize, is rapid cycling of genomic selection in maize biparental breeding works very well. I acknowledge all these persons that work in CIMMYT, all, you know, the CIMMYT Global Maize and Wheat, all the collaboration we have with Cornell through the Melinda Gates Foundation, with Jean-Luc, with Buckler, Mark, Jessica. Philomen, is Philomen there? Where is Philomen? No. Julie. And in Kansas, with Jesse Poland and Sarah. And these are the criminals of the biometric unit, some of the guys. But we were, you know this guy, he was visiting CIMMYT. And he was visiting CIMMYT, so he gave a seminar there. So this is part of the unit. So I think that with this, if you have any question, you can take other questions. There are so many, you know, so many issues that we didn't touch, but I think that we are learning, still learning all this.